A friend of David's and part of the IPD community. And so it's wonderful to be here with all of you today. I want to start off with just a quick acknowledgement of the team that put this all together, a creative, energetic team, including Ian Hayden, Rathika, Sylvia, Zari. I didn't see Zari here tonight. And Magnus and Jan, I think. And if you guys look around afterwards, I think that neon light in the back was a creation of Magnus. And then, of course, speaking about support, the support team has just arrived, and this would be David's family. So David is here with his biggest supporters, Hanala, his wife, Amanda and Ben, his parents, Marshall and Marcia, his sisters, at least one of them is here, extended family, and other family members. So I hope you'll get a chance to speak to them after this is all over, get some additional stories from the festivities in Stockholm. I wanted to also say that we're going to be hearing from some elected officials. So that's a strong show of support for this enterprise, and we'll see some in person and some with a recorded message in just a little bit. So let's wait for that. And I wanted to say that the IPD wouldn't be here at all without the steadfast support of the University of Washington. And it's been a partner and a great support for all the things that have been done here. I see Tim Dellett in the crowd here, who's the Dean of the School of Medicine. Some others might have seen here Paul Ramsey. Paul was a former Dean of the School of Medicine. It was really his vision together with David that helped put this together over a decade ago. So it's nice to see you, Paul. I want to say that there's also a great showing from the regents of the University of Washington. The regents hold a special place. They're the stewards of the university. They help guard the institution for years and intergenerations, and their wisdom here and their presence here is really a show of support for the IPD. And finally, I wanted to acknowledge the advisory board, who helps with David, helps guide some strategic decisions. With me here today are Clara Wu, who is here with us, as well as Rob Hirschberg, and Linda Stewart is signing off, but has been a trustee in the past, a board member in the past. So we're thankful to have them all. And I guess lastly, I'd like to say thanks to our partners here tonight, which is the National Nordic Museum. It's a wonderful venue. If you haven't been here before, I hope you've looked around. Obviously, it's a beautiful space here today for the lecture. So I want to introduce and ask to the stage Leif Carlson to say a few words from the museum.
Thank you. Sometimes doing things backwards can be a good idea. And I've heard Dr. Baker describe his work as doing things a bit backwards. And for us here at the National Nordic Museum, tonight is also a little bit backwards. I'm Leif Carlson. I'm the Executive Director and CEO here of the National Nordic Museum. And why this night is a little backwards for us is usually what we're doing here as the only pan-Nordic museum in the world is bringing innovations from the 5 Nordic countries here to the museum to expose an American audience to all the amazing things happening in the Nordics. Tonight, we're kind of going the other direction. So tonight, we're celebrating an incredible innovation here from our region that's being recognized by one of our 5 Nordic countries in Sweden. So that's a little bit unusual for us. We're also a little bit unusual in that we are the only national museum in Washington State but yet we get no national funding. Some of you may also be feeling the same right now. But we really are a community-led organization, and we're really proud of the support that we get from the community here in Ballard and across the state and across the country and across the 5 Nordic, as well as the semi-autonomous regions like Greenland, which has been in the news as well. So we do have a pretty broad view that we cover. So we go about 12,000 years into the past, and then we do a lot with contemporary Nordic culture as well. And so celebrating a Nobel is just perfectly in line with our mission here at the museum. So we're really, really proud of being able to have this event here tonight. We could not imagine it happening anywhere else but here at the museum. When people think about museums, they most often think that we're about the past, but actually we're about the future. The only reason that we collect the stories and the objects and the histories that we do is because we want there to be a better future for everyone. And while we're maybe content to kind of sit back and wait for that to happen, Dr. Baker's lab is making that future happen right now. And I think that's amazing to see. The amount of innovation that's happening there is absolutely incredible. And as someone who just days ago recovered from yet another respiratory virus, I'm very thankful that they're not slowing down at all. So with that, I would like to welcome all of you here to the National Nordic Museum for an amazing evening. It's my pleasure to have all of you here and to join our community. Thank you.
Thank you, Leith. Thank you, Leith. It is really a special time to be here and to be in partnership with the museum. We now have the opportunity to hear from some of our elected officials. First up is Governor Bob Ferguson. He's left a recorded message, and I just want to say quickly that I was very proud to see him at the rally on Friday, Stand Up for Science. Maybe some of you were there. But he made some powerful remarks. He's a great supporter of science in the state of Washington, and let's hear what he has to say.
Hi, everybody. I'm Governor Bob Ferguson. And first and most importantly, Dr. Baker, congratulations for receiving the Nobel Prize in Chemistry. Truly amazing and remarkable. Look, Washingtonians are, as I think we all know, among the most dynamic and innovative people in the world, and you're proving it. Now look, I am not a scientist, so I'll leave the technical descriptions of your advances to others, but I am the father of teenagers who take science classes and love them. So your success serves, I know, as a father, as an inspiration to them, and I suspect many other young people who are interested in the sciences. What I also know about you and your team is that you've accomplished what was considered almost impossible. Now, you once said, proteins are the workhorses of all living things. We recognize and celebrate you as a workhorse as well, and we're very lucky that you call the state of Washington home. So congratulations again on this significant honor. We all honor you and your hard work.
We all know Seattle's been a long-standing hub of innovation. And under Mayor Bruce Harrell's leadership, that continues to grow. I had the privilege last week of visiting a new initiative, the Seattle Climate Innovation Hub, that Mayor Harrell helped bring into this wonderful city. And as a backer of science and as a backer of progress, it's all of our honors to have Mayor Harrell here tonight to speak and to speak to David Baker.
Thank you, Brian. Just one sec. Let me get situated here. I have hardware here, right here, so I'm going to talk about that. I'm going to leave it right here. Now, who said computational protein design is not cool and sexy? I don't know who said that. I want to ground us in a quote as we celebrate Dr. Baker's incredible award, and the incredible pride we have in this country, but particularly in this region. The important thing is to never stop questioning. Curiosity has its own reason for existing. And that was Albert Einstein. Now, I just came from another. Where's Dr. Baker first? Okay, thank you. I was going to try to bluff it and try to work the crowd here, but okay, now I know who I'm talking to. Unlike our friend Bob Ferguson, I do have some speaking notes, and so I will slaughter some of this, but I'm going to try my best. But I have some good stuff in here, and I'll be quick, because at the end of the day, in a few minutes, I'm going to show you now. I'm not good at keeping secrets. None of you get one of these, but Dr. Baker gets one of these. That's where we're heading, okay? That's where we're heading. Okay. I was supposed to come at the end, by the way. Dr. Baker's contributions to computation protein design to turn I'm sorry. to turn amino acids into proteins earned him of course, the Nobel Prize in Chemistry, one of the highest honors in science. I should warn you at the top that, David, that unlike the Nobel Prize, there's no cash involved with this award. As director of the Institute for Protein Design and a pioneer in the field of computational biology, transforming the use of proteins to tackle some of our most pressing challenges, your work not only pushes the boundaries of science, but also directly improves lives across the globe. That's why we're so proud. We still want to be that global citizen in a world where we, as Americans, sometimes pound our chest. This kind of work defines us as the world citizen. Over 600 scientific papers published, shaping the field of biochemistry and influencing researchers worldwide. Rosetta software, developed by his lab, has been used by over 100 research labs worldwide to predict and design protein structures. Your team played a key role in designing COVID-19 antiviral drug and vaccine components. Let's give a round of applause of this science that allowed us right now to be here safely together. And the pride of University of Washington, one of the world's leading public research universities, consistently ranked top globally. Dr. Baker is actually the 8th University of Washington faculty member to be awarded a Nobel Prize. Their groundbreaking work spans in physics and medicine and chemistry. Annually, they receive over $1.6 billion in research funding, making it one of the largest federally funded research institutions in the United States. Now, in researching you personally, I was amazed at how much we have in common. We both walked the halls of Garfield High School, the historic institution. Councilmember Strauss, I know we're in Ballard, we're in your hometown, and I know I have Councilmember Sean Quinn here who hails in Tukwila area, but I don't know if there's other electeds here, but today we're talking about Garfield, okay? Just to get that. Okay, we share the Bulldog history, a deep connection. Both Dr. Baker and I grew up as child of government employees. In his case, 2 University of Washington professors, and in my case, 2 city employees. We both followed in our parents' footsteps. He carried on their legacies, Dr. Baker, dedicating his life to science and me to public service. And this is the part that really astounded me. We both can tell you about how deep learning models like RF diffusion can optimize generative protein structure design by progressively redefining noisy data. I don't know what the hell I just said. I just read it. Okay, we don't have that in common. But I share with you in closing that the key to the city represents everything we believe in. Selflessness, hard work, discipline, a love for people, a love for being curious. And somewhere along the line, someone loved you, and someone told you to work hard to serve others. And it is absolutely my honor to present to you, Dr. Baker, the key to the city of Seattle.
Well, it's hard to go after Mayor Harrell. We got some good comic relief there. We have one more elected official to hear from, and in a minute we'll hear from Senator Patty Murray. I just have to say that all you know, she's on the Appropriations Committee, and she is one of the fiercest defenders of science research and the funding of science research. She feels it in her bones, and it'll be our privilege to hear what she has to say. So, Senator Murray.
Good evening. I'm honored to join you in celebrating a truly remarkable achievement in science, innovation, and medicine, one that has once again put the University of Washington at the forefront of groundbreaking research. Congratulations to Dr. David Baker, Professor of Biochemistry and Director of the Institute for Protein Design at UW, on receiving the 2024 Nobel Prize in Chemistry. Dr. Baker's groundbreaking work has already opened the door to life-changing medical breakthroughs, including the first ever computationally designed COVID-19 vaccine that was developed at UW Medicine right here in Seattle. This is exactly the kind of scientific advancement that federally funded research makes possible. The National Institutes of Health has been the backbone of American innovation, supporting research that leads to cures and treatments and life-saving discoveries. Dr. Baker's achievements are proof when we invest in science, we invest in people's health and in our future. But let me be clear, these kinds of breakthroughs don't happen overnight, and they don't happen without strong, sustained funding for research. That's why I have always been proud to champion robust NIH funding in the Senate to ensure that scientists and researchers here in Washington State and across the country have the resources they need to succeed. Now more than ever, we need everyone to speak out and fight for the federal funding and policies that support NIH and critical research like Dr. Baker's. This is about supporting research and science, and when we do, we fuel scientific discoveries, job creation, and better health outcomes for all Americans. So, Dr. Baker, on behalf of the people of Washington State and the United States Senate, I want to congratulate you on this extraordinary and well-deserved honor. Your work has already changed lives, and I have no doubt it will continue to shape the future of medicine and biotechnology. Thank you.
Well, Senator Murray doesn't mince words, so we'll take them to heart. I just thought I'd mention, you won't see too much more of me here tonight, but I wanted to mention a few things, especially for those that haven't been to Stockholm, especially during the Nobel ceremony. There is a tradition there that the Nobel laureates leave behind a personal item. And for those you're not aware, David left behind a broken ski pole. And for those that know David, they know that carries some significant meaning. In particular, resilience, adaptability, and as Senator Murray mentioned, there is some threats to the work that we all do here in the IPD community and the university writ large. So it's something that I think we all think about and we all take seriously. I think David wants to make some remarks in this area, so I'll turn the mic over to you, David.
Yeah, well, first of all, I'd like to thank all the people who made this event possible, and in particular Brian for running the show, and he's just been a wonderful friend and supporter. But in having this celebratory event, I thought it would be important to also comment on what's currently happening as it relates to science. And I think everyone here appreciates the importance of science, so I don't want to be depressing, but I want to give a description of the facts so that when you're talking to people who aren't scientists, to friends and relatives and people in other states in particular, just to give you some things to some things to tell people about. We've had many conversations in my group about how do you deal with situations like this, and I think the most important thing to do is try to act and try to communicate what's happening. So, and again, what I'm going to say is well known to some of the people here, but let me just briefly tell you how the sort of innovation, how innovation arises in biomedicine and where drug discoveries come from. A large percentage of the innovation is made at universities by graduate students and postdocs, who then often take their ideas and spin out biotech companies, and the biotech companies further develop these, and they are eventually, and the ideas eventually transition to pharmaceutical companies. So it might be surprising to many people that actually most of the innovation doesn't actually happen in the big companies. It happens at universities, and the universities also train the workforce for the biotech and the pharmaceutical companies. So, there's this idea that, maybe some uncertainty and the threat of, and cutting funding can lead to sort of, maybe revitalize a company, but I just want to, my main message tonight is that exactly the opposite is true for science, and that's because of the critical role that graduate students and postdocs play in this. So, first of all, in face of uncertainty about funding, universities are cutting way back on graduate school admissions. So, for example, in my department, the acceptance rate, the number of students admitted has gone down by more than a factor of 2, and that's true across the country. So it's going to be much harder for students next year who want to pursue careers in science to become scientists. Next, because of the way in which program managers have been fired, there's a lapse in renewing the flow of funding. So labs are stuck without funding, which makes it very, very hard for the graduate students and postdocs in research labs. Now, because of the uncertainty, both universities and universities are imposing hiring freezes. That's happened at the University of Washington and the School of Medicine, which is a very reasonable response because we don't know where their money is coming in. So that means that that new postdocs can't be hired. It's also individual labs don't know if they're going to have funding. And then finally, for postdocs transitioning to become faculty members, faculty job searches are being canceled across the country, again because of this uncertainty. And so the cumulative effect of this uncertainty is way out of proportion to the amount of funding saved. Because it doesn't actually save that much money to have all this uncertainty. It just wreaks havoc on a system that has to behave conservatively because it can't pay people if there isn't money coming in. So there's a final aspect to this, which is, as you know, many outstanding researchers come to the United States from foreign countries, and with this uncertainty, it's becoming increasingly, the U.S. is getting increasingly less attractive as a place to stay. So people are having to return to their home countries, and so the U.S. is, U.S. is losing out on all this talent. So I'm telling you this just so to give you a few more talking points with people that you're speaking about, because I think these things could really have a very long-term effect, negative effect, really not just in biomedicine, but across science overall. And so again, my message, the bottom line is that uncertainty is really bad for science because it hits people at the most, the most delicate time in their career, early on, like undergraduate students wanting to become graduate students, or graduate students wanting to become postdocs, or postdocs wanting to become faculty positions. So anyway, so that's the message to spread, and now we'll move on to more positive things. So.
Well, we've heard a few of our challenges, but as David said, it's now time to turn to a more optimistic note. And I was going to do this earlier, but I have the pleasure to introduce the Nobel lecture, David Baker, and his lecture, which he'll about to give. I think we heard from Mayor Harrell and others. Some of and everybody here knows David quite well. I mean, there's tens of thousands of citations for the papers he's written. There seems to be a new peer-reviewed publication, every month or two. His students have gone on to really profound and reputable careers as scholars and as entrepreneurs, many entrepreneurs here in the room tonight. So it's quite incredible. But perhaps a little bit what I would share tonight is a taste and a few observations from Stockholm and the wonderful event that we had in December. For those that didn't follow it closely, it occurred in the middle of December, which is a cold month in Stockholm. We were greeted by a dusting of snow as we arrived, and we had some crisp, clear nights. The latitude of Stockholm is 60 degrees north, and nearly 60 degrees north, which means that the sun sets before 3. We thought we had short days here, but Stockholm has very short days. The Swedes, to their great ingenuity, have developed countermeasures over the years, and one of those is city lights. So we are all regaled by the wonderful Nobel lights, really works of art, all scattered throughout the city, which was wonderful to see. What was particularly unique this year was the showing from the IPD. So the IPD community showed up in force. We had nearly 200 individuals that descended from all around the world to support David in his ceremony for the Nobel. We took the Museum of Modern Art for the night before the big ceremony, and had music, had great food and warm conversation throughout the whole community. I saw people that hadn't seen each other in years and maybe worked together decades ago in David's lab, sort of have wonderful conversations. So it's part of really David's character to bring people together, and that was a quintessential moment for that. We also had some interesting the night after. This is the formal ceremony, so it's a chance for all of us, for me, first time ever to wear a white tux, which means tails. The women were in full, wonderful gowns and dresses, and so it was quite the affair. David's family was there in full force. Brian Coleman was there, one of David's early collaborators, Lance Stewart, Tricia Davis, Neil King. So it was a wonderful support crew for David. And what some people don't know is that actually David and Hannela had the seats of honor at the banquet. David sat next to the Queen and Crown Princess, and Hannela sat next to the King of Sweden. And many of you know, but Hannela is Finnish, sitting next to the Swedish King. So I heard there was some spicy conversation that evening. So now it's time to turn the mic over back to David and to hear some of to learn and to hear what was said and what's been going on at the IPD. So David, let's start with the lecture. We're all here to learn something.
All right, well, thank you again, Brian, for all that you've done, and thank you all for being here. So today I'm going to tell you about proteins, and I'm sure there are many things that Bruce could tell you about that I don't know about, but I'm not really prepared to discuss them. So other than that, we could compare the old Garfield to the post-renovation Garfield, which my kids can both describe. So today I'm going to talk about de novo protein design. So proteins are the miniature machines inside in all living things that carry out essentially all the important jobs. So when we eat, proteins digest our food. When we move, our muscles are made out of protein. They're powering the movement. And basically everything that goes on in living things, including capturing energy from the sun and turning it into making molecules. So, and these little blobby things in this picture are an artist's rendition of the proteins in a cell, or a small subset of them. So as I said, proteins carry out all the important jobs in living things. And where did proteins come from? Well, humans didn't create them. They evolved by natural selection over billions of years to solve the problems that were relevant during evolution. But the problem is that, and so proteins can do amazing things. I mean, we're all, everybody here is a testament to the amazing things that proteins can do. And if you go to, you look outside at the plants and animals, it's all powered by proteins. But these proteins, they didn't evolve to solve current day problems, and we face many. So that we're heating up the planet, we're polluting, people are living longer, so there are new diseases like neurodegenerative diseases that are becoming very important. And for at least some of these problems, if we wait another 100 million years or 1 billion years, proteins might evolve to solve them, but we don't want to wait that long. And so what I'm going to tell you about today is our new ability to make up brand new proteins to solve current day problems. So first I have to give you a little bit of a primer on biology. So we all have genes in our genomes, which are made out of DNA. And what those genes do is they're instructions for making proteins. And each gene describes the sequence of amino acids in a given protein. And that sequence of amino acid folds up into a structure, and that's the machine, and that machine carries out a biological function. So in protein design, we go backwards. We start off with a new function that doesn't exist, like curing neurodegenerative disease. So we think, well, what kind of protein could be possible that could really have an impact on this disease? And then we think about what biochemical functions could help, like maybe destroying some of the proteins that are implicated in neurodegenerative disease. And then then we make up a brand new protein on the computer that we predict would have that function. We work backwards to find an amino acid sequence that would fold up to that structure. And then we go backwards from there to use the genetic code to come up with a DNA sequence which encodes that protein sequence. So at that point, I told you that we have genes in all organisms that encode the proteins. Now we have this new gene. It doesn't exist anywhere in the world because it's a brand new made up protein. So what we do is we take that new gene, we actually make it in the lab, a piece of DNA that encodes that new gene, that new protein. We put it into bacteria, and the bacteria make the protein, and then we can get the protein out and see whether it actually has the function that we wanted. So this is interesting for a number of reasons, but one of them is that there's a huge number of, the space of possible proteins is absolutely enormous. So, the proteins that we know about in nature are a tiny, tiny fraction of all the proteins that could exist. And so previously what people did when they wanted to try and make a protein do new things is they took a protein that already existed in nature and changed it a little bit. It's like breeding a new kind of cow. We instead when we want to make a new protein, we search the whole space. So we're building it completely from scratch to do what we want to do. Now we come to the part that Bruce was explaining to you. So how do we do this? Well, in the last 5 years, we've been developing AI-based methods to do this, and they're inspired by methods for generating images. So, the way that image generation AI methods work is you take a very large number of images off the internet or somewhere else. You add different amounts of noise to them, kind of shown across the top here, and and then you train a neural network to remove the noise. And once that's been done, you can start with a completely random collection of pixels, like shown here, and if you progressively denoise it, you end up with a new image that looks like it could be from the internet, but actually it's completely new. And in the same way, we took all the protein structures which are known, which have been determined by experiments. We added different amounts of noise to them. We trained a neural network to remove the noise. And once we had done that, we found that we could start with completely random collections of amino acids residues. That's what's shown, sorry, it's hard to move this, shown on the bottom left, and then progressively remove the noise. And this resulted in brand new proteins, and the animation on the left shows you the course of one of these calculations. And what this assembles into is a brand new protein that looks like the proteins that have come through this natural evolutionary process, but really it's completely new. So you might say, well, what good is it to just be able to make up new proteins? And where it gets interesting is just in the same way that you can make up a new image, but you don't have much control over it. With images, you can provide a text prompt. You can say, generate an image of a cat sitting on a table, and that's what it will do. Well, in exactly the same way, we can tell this protein generation process called RF diffusion that we want to generate a protein to bind to a protein involved in neurodegenerative disease, or in the case shown here, to the insulin receptor, because we're interested in developing improved forms of insulin. And so what happens is the insulin receptor is shown at the bottom here, and now this sort of random diffusion process is happening, and what's generated is a protein that looks to someone who's an experienced structural biologist like it should bind insulin, but it's completely new. It just has all the properties that you would expect for a protein that binds to insulin. And so you can see its shape is roughly matches the shape of the receptor. So then what we do is we find an amino acid sequence which encodes this protein. We make a synthetic gene and we put it into a bacteria. We get the protein out and we measure whether it binds the insulin receptor. And in fact, Jin Ruang, who the postdoc lab was working on this, has actually made a number of very interesting, potentially improved versions of insulin using this method. So today I'm going to tell you about applications of this technology in medicine, technology, and sustainability. And I don't have very much time. I don't want to keep you from the party, so I'm just going to give you a couple examples in each area. So a first example will be a second example beyond insulin of designing proteins to bind to other proteins is the design of proteins to block snake bite related injury. So snake venoms are toxic because they have proteins in them, this gray protein down here, which when they get into your body really cause a lot of damage. And so Susana Vasquez-Torres designed a series of proteins which block different components of venom. And our collaborators, and I should say everything that I described is very, very collaborative. This was with a group in Denmark, showed that if mice that effectively got bitten by a snake didn't do very well, but when they got the antivenom, they were completely protected. And so we're very excited now about developing this as a general antidote for snake venom. So the current state of the art is horses are injected with snake venom, and then they collect the serum from those horses, and then that's sent around the world to be used to treat people with snake bite. But you can imagine it has to be kept cold. It's difficult to make, and so this could be a much cheaper, much more distributable form of an antidote. So we've been very interested in developing improved medicines for inflammatory disease and for cancer and chronic disease. So this is in gray here is the TNF receptor, which is at the center of much of inflammatory disease. And many of the drugs that you've heard of that for rheumatoid arthritis target this receptor or the ligand, compounds like Enbrel or Humira. And so here you see a diffusion trajectory where we've just given it the receptor and it's generating this protein that kind of fits perfectly against the receptor. This protein binds very tightly to the receptor, and in models of animal models of inflammatory disease, this protein is looking very, very effective at suppressing inflammation. And in the cancer area, there are a family of molecules called cytokines that our bodies produce that activate the immune system to fight pathogens, and they've been used to attack cancer, but they're problems. They haven't really been the proteins our bodies produce are not really optimal for that purpose, and so we've made improved versions, and this shows very encouraging results with pancreatic cancer with one of these improved molecules, an improved de novo designed version of IL-21. And on the topic of respiratory disease that came up earlier, we've been designing proteins against influenza and during the pandemic against COVID. But I put this example in here because I wanted to show you that we can condition this process more than just by giving the target. So what we can also say is what sort of protein we would like. So the protein that's being generated here is actually an antibody-like protein, and this is important because the pharmaceutical industry is very much favors using antibody molecules as medicines. And so now basically what we're finding is we can now design antibodies completely on the computer. The way that they're obtained now is by immunizing an animal or trying to identify the animals and the antibodies in individual people. And just to show you how well we can do this, this is a blow-up of this region here. This is the structure of the influenza surface protein, and if you compare the computational model to the actual structure, which we can determine experimentally, they're almost identical. And then finally, coming back to neurodegenerative disease, we have been designing proteins in one of the approaches that bind to some of the major components that are found to accumulate in the brains of Alzheimer's patients. These are amyloid structures made out of A-beta or tau, and we're very interested now in approaches that will systematically destroy these fibrils. And what I'm showing here, this is great because my colleague Thomas Knowles, who I took skiing yesterday, is actually in the audience, and his group showed that these designs completely block, he's in Cambridge, England, that these completely block the formation of fibrils, both of A-beta fibers and tau fibrils. So those are a few of the examples in medicine, and now I want to shift gears and talk about applications outside of biology. So one of the things that proteins do very well is to sense. So the olfactory receptors in our noses are proteins, and basically everything that biology uses to sense the environment is a protein. And so it stands to reason that one should be able to build new and improved sensors. And so one of the ways in which sensing works in biology is by modulating the electric current that goes through proteins called channels. And so we were interested in sort of generalizing this concept. And so the first thing that we did was set out to make a series of channel proteins with different diameters. And these channels are inserted into lipid membranes, as shown here, which can't conduct current. And so what we find is that when the diameter of these channels gets wider, their conductance increases. There's more current going through. So now we're very well set up to make new sensors. So we can take proteins that have been designed to bind a small molecule and that we want to sense, and put them at the top of these channels. And then what happens is that in the absence of the small molecule, this channel is open, so current can go through. But when the compound that that's been designed against is present, then it closes up and the channel completely, the current can't go through anymore. So there's a very clear electrical signal of whether that compound is there or not. So that was in membranes made out of lipids, but we've recently been able to generalize this to work with silicon nitride, which is what computer chips are made out of. So here we have a silicon nitride chip with a hole in it, and I'm showing you pictures here of designed proteins which have exactly the right shape to fit into these holes. And here are a bunch of different shaped pores that can shape proteins that can fit into these pores. And these beautiful structures are made by Alexis Courbet and Jinwei, and Jens Gunlock's lab at the UW has shown that these actually will insert into silicon and give very stable conductances, which is a very exciting step towards really marrying biology and electronics. So you can imagine your cell phone could suddenly tell you what chemicals were in the environment, or for sequencing applications like protein or DNA sequencing. So one thing that is very important in biotechnology are things like gene editing that you've heard about, the ability to design proteins that will, the ability to target very specific sequences of DNA. And there's been a lot of excitement about CRISPR-Cas9 for this, but it's very, very big, and it's not as specific one would like. So we've been designing proteins that bind, and here's a picture. This is the DNA and this is the protein. They bind in such a way to be able to read out exactly what the sequence is at the protein-DNA interface. And these designed proteins can have very high specificity for their target sequences. And then I mentioned, or it was mentioned earlier, that my colleague Neil King at the Institute for Protein Design developed the first computationally designed medicine, and that was a COVID vaccine. And that was based on designed self-assembling nanoparticles that Neil created. And then what he did was to take the spike protein from the COVID virus and put part of it on top of the particle. And then when you get injected with his vaccine, you make a huge amount of antibodies against the particle. And this is our first medicine that has been approved for use in humans. So now the problem with this is, no, this is very, very good. There's no problem. But if you want to make these particles bigger, it was one of the things that is very interesting, that often you want to get drug compounds into cells. But those drug compounds, I mentioned CRISPR-Cas9, can be pretty big. So what you'd like is to be able to make a big container that you could use to get compounds into cells with. And so I showed you the containers that Neil made, and we wanted to make them bigger, but it turns out that there's no way to make them bigger without sort of disrupting some of their nice properties. So if you think about a structure that's made out of pentagons, this is basically a dodecahedron. It's got 12 pentagons, and it turns out there's nothing bigger that you can make that's basically the same on every side. That's all pentagons. But you all know that you can, if you take something like that and you insert hexagons, you get something that looks like a soccer ball. And you can keep this process up. You can keep adding hexagons. But there's a problem from the design point of view, because now different locations are different. So, for example, this edge is the edge of a pentagon and a hexagon, but this is an edge just between two hexagons. So we have to design proteins that can be, we can either design a protein that can have two different, can be in two different places, even though it's the same protein. It has to have slightly different shapes in those places, or we have to be able to make these structures using multiple proteins. And these are those two solutions. These are actually experimental data on structures determined of particles made using just one type of protein that adopts different kind of geometries in different places, or several different types of proteins. And for scale, this is what's currently used for gene delivery, AAV. And you can see you can package a lot more in these type structures. And then finally, everything I've talked about so far is kind of static. But in biology and in many applications, you want something like that has switch-like behavior. So you can, for example, turn it on and off. And so we've made a lot of progress in designing proteins that can be switched on and off. So here is, for example, a protein that sort of looks like a hinge. In fact, that's what we call it. So it's a design protein that when this circular compound comes along, it bends in this way. So if we make a triangle out of this protein here, and we add this small molecule, then it changes shape and adopts this square-like structure. And so this works very nicely. And this is just sort of a proof of concept to see if we can make switch-like systems. So here, when we add the circle compound, this is electron microscopy data. It goes from being a triangle to a square, here from a square to a pentagon. Now, you might say, what use is that? Well, we can actually use this to make medicines that can be turned off. And so here is one of the cytokine-like molecules I described earlier. It's a mimic of the cytokine IL-2. And now here is the equivalent of that circle compound I described. That is, when it binds to our designed IL-2, it changes its shape. And what I need to tell you is the way this IL-2 works, the way it activates the immune system is by bringing two receptors together on our surface of our cells, this red one and a blue one. And what happens, you can see when this binds, is it causes this change in shape, and that kicks off the blue component. Well, this really works. Our collaborators in Germany can actually follow what happens to the red and blue receptors on cells, and they see when the first compound is added, they come together, and then when the off switch is added, they come apart. And you can see that here. This is the amount of you see the receptors kind of flying apart after this off switch is added. And so what goes along with this is that the signaling produced by the molecule shuts off very rapidly too. So this gives a way of modulating the amount of signal and the types of signal that a drug molecule produces. And then finally, in the last few minutes, I just want to tell you about some applications in sustainability. So we've made a lot of progress in the last year in designing proteins that catalyze chemical reactions. That is, that either promote the making of bonds or the breaking of bonds. And so basically what we do is we start with just a description of a specific part of a protein, amino acids, in a position that we predict would mediate a chemical reaction. And then we use that same diffusion approach to build up a protein around it. And so we can build up proteins with different types of reaction centers. And we can experiment to see what geometry of and what placement of amino acids gives what type of activity. And in doing this, we've been able to make very, very active enzymes. This is exciting because, and we can actually determine experimentally how these enzymes work. And what we find is that when we are making up these catalytic sites, that we can actually reproduce, that they actually come out almost exactly as we've designed them, with the side chains in the right places. We've been able to make proteins that break down, that carry out other chemical reactions. And just to give you some color as to how this all works, in this experiment, we made 96 designs on the computer, made 96 genes, and those 96 genes we then made 96 proteins from, and the best design was, as in the case I showed you before, was very, very active. And actually, Seth, who did this experiment, just pulled me aside just before I came over here to show me that he has his latest run of testing a brand new set of designs leaves this one way, way behind in the dust. So it's an exciting day. But this, when he first got this result a few months ago, was very exciting indeed. So where this is going is we can now take this basic concept of how to break chemical bonds and build it into proteins that we're designing, not to bind to the small molecule that was relevant here, but to polymers such as plastic. And we have our first results now showing that we can design proteins that break down some of the polymers that are in plastic, which is very, very exciting. And so we're excited to see as we improve are able to improve the activity of these enzymes, we're excited about being able to make a dent there. Other problems that we're very excited about there are we can design proteins that form chemical bonds. I'm not showing you that, but we're excited about new routes to antibiotics and other types of drug compounds. And then one of the things I mentioned earlier that nature is really good at is capturing sunlight and using the energy to make molecules. And so that's done in plants by a very complicated system called the photosynthetic chain, and this is the photosystem. And that's done by the key there is a pair of chlorophyll molecules. That's what make plant green. And so what we've done is to design proteins that hold two chlorophyll molecules in exactly the right same arrangement that they are in the photosystem. So you have this huge complicated protein, and now we can make just a very simple protein that holds these two chlorophyll molecules in exactly the same location. And we can use that now as a starting point for building artificial photosynthetic systems and for enhancing natural photosynthesis. So here's an example now of taking that protein that had two chlorophyll molecules, that bound those two chlorophyll molecules, and building it into a higher order assembly shown here, which should be better at, increasingly effective at harvesting light. And so we're now building components out to increase the amount of light that's captured, to change the spectrum of the light that's captured so we can enhance photosynthesis in plants, and to couple this to chemical reactions. And so a final example of where we can go far beyond what nature has experimented with or discovered. Some of the remarkable materials in nature are things like tooth and bone and seashells, which are proteins that are templating the deposition of inorganic minerals like calcium carbonate and calcium phosphate. And so we've really been intrigued by this, and what if we could learn to design proteins that could template the formation of new types of materials that were, have some of the remarkable properties, but have other of the biological ones, but have new properties that nature didn't really care about. And so, for example, we've had success in templating the growth of zinc oxide, which is a semiconductor. And so here we do that by designing an interface between the protein that should organize the components of zinc oxide to form in the regular repeating lattice structure of the zinc oxide crystal. And so we've put that interface into these cage-like structures here, and if we put these into zinc oxide, we see that region filling in with zinc oxide. So we're very excited now about this as being a new route to building semiconductor materials from the ground up. So I've given you today examples of the application of de novo protein design in medicine, in technology, and sustainability. And we really are just at the beginning, because I think we've collectively just sort of figured out how to do this, and the number of sort of possible applications we believe is really, really broad. And so really the key to all this is all the amazing people who've contributed to this work over the years, many of whom are here, and it's just such a privilege for me every day to get to work with everybody. They're all half my age now, but so I have the best job in the world. Again, thank you for coming. I want to thank my family for their love and support for being here. I want to thank everyone for coming, all the students and postdocs who made this possible, the people who organized this, and the Nordic Museum for letting us use this beautiful place, and Brian again for emceeing. So thank you.
Wow. I've got a little ache in my knee. Maybe the IPD will get some bone growth there to help me out. But that was just a great vision that you set forward, David. Let's please all stand and give David a well-rounded, robust applause. Okay, we're at time, so I'll try to be quick here. I just want to say to everybody here that we're really in the middle of a journey. The recognition by the Nobel Prize Committee is for work done, but as David said, we're at the precipice of many, many new discoveries. So we hope that David and the team at the IPD accelerate their good work so that we might even benefit from some of the discoveries and innovations that they have. We're going to move now, and I would ask everyone to stay seated for a little bit. We're going to empty out the first couple rows. I think there's some ushers that are going to help us, and then we have to clear because the next group is coming in, which is David's current students and former students, so there's a big party happening. You're welcome to enjoy the Fjord lobby and have conversation, but this concludes the event. Thank you.